Good afternoon. I am Pei Ping on behalf of Dr. Carol Watson at UCLA, the Bridge Center for Skills and Workforce Development Lecture Series. We welcome you to join us today for one of the most fascinating lectures in the large language model lecture topics. We welcome Professor Jake Chen from UAB, who's the founding director for informatics centers at UAB, and who's a professor of computer science, human genetics, and biomedical engineering. Jake will guide us through his own research on the translational values applying large language models. Jake's research over the years has focused on AI and ML research and has been a leader in multiple biomedical communities with strong application of machine learning and AI applications. He is so accomplished with a collection of recognitions from the community and also assuming broad leadership roles in multiple scientific relevant organizations. Today, Jake has crafted lectures to guide us understand the challenges and opportunities on developing derivative models in addressing genomic and systems biology research questions, to understand the basic concepts and knowledge representation techniques for characterizing biological processes, molecular function, and experimental perturbations, all the way linking them to phenotypic impacts, and to understand the current research trends in apply AI, ML, BERT, and large language models for translational relevant biomedical questions. Welcome, Professor Chen.
Thank you, Professor Ping, for the very kind introduction. Let me just share my slides and make sure that everyone can see it. All right, so I think I also have a floating meeting controls. So I'm really excited to be able to share some of my curiosity toward this topic, as many of you are also working very hard in studying, understanding what's going on in the generative AI models, and how can they apply to biomedical research. It could be basic research, could be translational research. So this is really not a recipe for success because we're just starting the revolution. We're right in the middle of it. But I hope that I give you enough pointers so that you can find tremendous research opportunity and go back and contribute to the field. So to begin with, I would like to mention the basic concept. What is a generative AI? It's a form of AI in which models are trained to generate new content based on natural language input. So essentially, it is expected that somehow we give instructions, the AI model would understand it. And if we throw the AI model with a variety of biological data input, for example, we may be very interested in genomic information. We'll give DNA sequences, gene expression information, and additionally, we may also give it patient electronic medical records, imaging information, pathology report, socioeconomic data, everything that we throw at this model, and we're hoping that the model will actually generate new actionable biological hypotheses that we can act, right? But today, I don't think that the thing existed. There is something approximate. I think there's something that everyone have become very familiar with called the ChatGPT exist. I actually had the fun of just entering into the ChatGPT prompt of today's lectures, right? I said, well, here's me. I'm going to give a lecture on generative models on December 14th. And then there's a bunch of learning objective. And please generate a Chinese translated paragraph to promote the webinar for Professor Jake Chen. So in a sense that this is a generative AI. Let's see how ChatGPT generated. So ChatGPT said, well, certainly, here's the English translation. Join us for a cutting edge webinar. He's not very humble, but the cutting edge webinar on this topic by Professor Dr. Jake Chen. This series will feature large language models and their pivotal role in genomic and systems biology research. Mark your calendar. I didn't say calendar. So the generative AI is able to fill in the void and sound like a very promotional and be part of the innovation that is reshaping the future of bioinformatics. But those of you who read Chinese, you see that the Chinese translation is also pretty good. Right? So it said 加入我们成为重塑生物信息学未来的创新之一. That's pretty good. Except that I found that there's maybe a slight misuse of this term 网络. It should really be 线上 or online, not network webinar. So I would deduct 0.1 to give this generative AI text 4.9 out of 5. Okay. So how about generating even further a poster? Now, many of you have done poster and you have seen the poster for my presentation today. It contains images, text, and some kind of a reasoning how to put the text and image together. So I said all of the above. I'm very excited to say, well, would you please generate a catchy poster to promote this event among biomedical data science trainees? And here's what it generated. On the surface, it looks pretty good, very impressive, but I cannot read any of the text. It's very busy, very colorful, and the text doesn't make sense. And I was somewhat disappointed. And I said, well, please keep the poster minimalistic, interesting, and highly relevant to the textual description provided, and regenerate. Do you think it does a good job? Let's see. It's certainly cleaner, but it misspelled bridge to AI, and it doesn't seem to understand bridge to AI in the generated figure. It does show some trees, but when you look at the date, it doesn't make sense. It's garbled. And I was quite disappointed. And for the third and the last time, I said, please make sure to use fonts of the same style and real English text if you show them on the poster. Right? I was demanding to keep the image, color, and text styles consistent and minimalistic. I even put the prompt according to proper prompt engineering to be very polite and say thank you. And here's what it generated. It does look better, and there's textual portion and image portion, but somehow I was continue to be disappointed as all of you are now. So the generative AI is here, but it's probably in its nascent state. If it cannot generate a poster well enough with my straightforward prompt engineering method, and how can we expect it to excel in such complex tasks of translational biomedicine, right? So I give it 2 out of 5, and maybe I was lenient. But if you look at the generative AI model according to Google Trend, generative AI as a search term wasn't popular until only about 18 months ago. And I would say that mostly it's driven by the ChatGPT 3.5 launch. In fact, if you go to PubMed and search for generative AI models, and there are only about 1,700 papers, and many of them are not actually the hardcore generative AI model. They're AI machine learning method that will generate some labels, but they're nonetheless marginally related to the canonical definition of generative AI. If you go to Hugging Face, however, you do see many generative methods, machine learning methods, that's already popular, and there are 1,800 of them. If you go to GitHub, there are actually a lot more projects that are claiming to use some kind of a generation, mostly text generation, of course, their image generation. And I would say only a few percentage of them are related to biological sciences. So it would be good to actually systematically look back to see what is the history of generative AI. According to Gartner, which is a business intelligence company that provide comprehensive reports for the business commercialization community to act on. Generative AI really started about 2010 in the near perfect translation of natural language. So in this large language model series, rightfully expert who have talked about how the large language model or generative AI model has been applied throughout the years to biomedical texts, which are mostly physician notes or PubMed articles. And then it progressed to gradually understand the meaning of words, primarily through contextual learning. And then as the model become bigger and people have found that the performance usually become better, there's an emergence of intelligence that come out of these large scale models. And then over the years, people realize that we also cannot have isolated models for specific tasks. It would be better to generate so-called foundation models, such as the GPT and LAM. And then those are real conversational in style. You can use human languages, not necessarily any programming language to achieve the effect. So to the point that people are predicting, Gartner specifically are predicting that today, there are actually 0 AI generative drugs. They're predicting, I'm not sure whether they're too optimistic, they're predicting that 30% of new drugs in the future will be generated by AI. Certainly, I can see that for biologics drugs that are complex antibody structures, people are already using AI methods. For marketing, and if we're not already inundated by marketing messages in the future, Amazon already is implementing AI-generated keywords to say what people are saying about a certain product, the pros and cons. But in the future, more than 30% of those messages are going to be generated by AI, for good or bad. And then the future trend across the industry are 40% of enterprise applications will have some kind of embedded conversational AI. So that's not surprising that today you hear that the in hospital information systems. You're increasingly seeing physicians adopt large language models and being able to dictate the notes and share and communicate with other insurance companies or patients. And 30% of enterprises will have AI augmented development and testing strategy, and by leveraging on the reasoning capabilities. And 60% of the design website and mobile apps will be AI generated. More than 100 million humans will engage robot colleagues at work. And the robot colleague doesn't necessarily take the form of the hardware robot. It could be the software agent that serve as assistant, physician assistant, or my research assistant in the future may be a robot. And so many of us will have to deal with, engage the robot colleagues because of the generative AI advancement. And then even 15% of the pure AI generated applications will be just completely free of human. So the world is changing and very rapidly. So that's what we're dealing with, to the point that the Nature just a few days ago, actually yesterday, just came out with the Nature's 10 people who have helped shape the science in 2023 and named one non-human. And you may have guessed it. It was ChatGPT. It's the star of the year in science. So ChatGPT is in the spotlight because it can assist researchers in writing articles. And we've seen Professor Liu mention that she prepared a part of her slides for the series using ChatGPT. People are, professors are finding that ChatGPT is so easy in parsing out articles and even help us write papers and grants. But then it does raise concerns. What about the confidentiality and so on? I won't elaborate at this point. It does help us perform mundane tasks we usually delegate to research assistants. It does help us clarify our ideas. A student could misuse it. For example, if the students are trying to pass through exam and they somehow use this ChatGPT, it would be considered the same as cheating for maybe sixth graders in the math test, and then they pull out a calculator, and it's really not good. And then the AI, ChatGPT is certainly biased. We know that when we don't fine tune it and train it for biomedical text and knowledge, it would certainly have biases on common information that's on the Wikipedia or in the public domain that may not represent the truth, but simply may represent the dilemma or the common consensus that the science is trying to overcome. And it has questionable environmental impact. Imagine that if you train the ChatGPT with tens of thousands of GPUs that cost just a tremendous electrical power, when a graduate student only have a brain of maybe 30 watts of power for a day, and then the energy footprint of human is still considerably much better than machines. And then the lack of transparency is even more severe in science. We do want to be able to find out how ChatGPT is arriving at a conclusion. It does better in mathematics than in complex biology, whereas the details of science is simply not in the vocabulary of ChatGPT. So we talk about the science, and especially I talk about the translational biomedical research. I want to give you a perspective of what I meant by this central paradigm. The central paradigm can be exemplified by this diagram I showed for translational oncology research. In oncology research, people really want to find out novel cancer drugs or combinations that can be applied to different individuals, or biomarkers that can test which people may benefit from these drugs. On one side, and because of the National Institute of Health funding, there are tremendous data from TCGA and many other similar projects. They've been producing multimodality big data that captures every aspect of the patient, including the patient's clinical history, the tumors, description pathology, the imaging information, the genomic information of the patient, the normal tissue, the tumor tissue, and the microenvironment, including the immune profile of the patient. So all of this information are moving from left to the right side with the initial idea that some kind of a mathematical model would be able to help address it. And then the mathematical model is going to be imprecise. So hopefully we'll couple it with a self-learning healthcare system. We'll try to introduce some drugs, some practices, and we test it on cohorts. We do very fast clinical trials, and we see the outcome. And then hopefully through more and more trials, and we learn about the drug, and then eventually some drug will eventually succeed, and then patient will benefit. In this paradigm, I would like to point out that we always have to think about primarily three types of data in translational medicine. There's gene that capture the genetics of the patient. There's a disease that's at the phenotypic level that connects to the electronic medical records and then ontology. And then there's the drug that generally is considered as intervention. Of course, and you can also say that there should be environmental factor. But then if you broadly think about the drugs and environmental factor together as perturbation, and this is still applies. On the model side, the translational medicine really figured in the traditional way is going from cell lines to animal models to humans, right? So there's no place for in silico models yet in the bulk of the translational biomedical research. So for example, this is a figure from a paper. The size of the dot indicate the subject domain, and there's an arrow pointing to the shift of a translational path of paper citing the domain and where they're actually coming from in the three, just a triangle of this translational biomedical space. So as you can see that the bacteriology is more done on the cell lines. Veterinary medicine is more relying on animal models. Rightfully, nursing is only for humans. All of these other are moving in between. So this is a translational research landscape. And the question is, can generative AI today already making some impact? I actually recorded a 3-minute video, and let's play it. Imagine I'm a person who does not know too much about immunotherapy. And I'm asking the question, what are the hottest targets for immunotherapy today? It's giving me all of these answers, and I'm interested in seeing immune checkpoints. I already know some. And interesting target, TIGIT, that's known to inhibit immune responses during viral infections. And tumor-associated antigen, that's for cancer therapy. Let's say I'm very interested in TIGIT, and I said, can you tell me how to obtain the protein structure for TIGIT? now the ChatGPT is giving me some vague answers and saying that there are different ways of getting the structures. The best way is to go to PDB. I've decided that I want to be lazy. So show me a table summarizing the PDB ID of TIGIT in human, their derivation methods and date of publications, and their structures. Wow, there is. There are altogether 5 structures. They were all published last year with the TIGIT in complex with poliovirus receptor. All of these structures seem to be good. Of course, you need to verify it. And the way that I want to verify it is to write a Python script. But suppose I'm starting to learn Python and I want the software to show me Python script. And wow, it does show a Python script that will pull these IDs and query the PDB database, retrieve the results. Now I'm still not happy. I want to develop a drug against a TIGIT target. And I want to find out what has been done. So GPT is telling me that there are monoclonal antibodies that's been in clinical development. But can you tell me antibody design tool that can design antibody de novo? It gives several software tools, including the very famous Rosetta antibody design tools. When I continue, I said, well, can you show me how to write a Python code to use this Rosetta antibody design software to design antibodies against the structure? And then it showed me the Python code and 10 models right away that I can start testing. Okay, so I don't know about you, I was very impressed because in 3 minutes, I was able to overcome the difficult learning curve of so many different disciplines, from genetics to immunology to protein engineering and data science, right? So very few of us without proper training would be able to do it. And so if you're not convinced that the generative AI model is here to really help, here's another example, right? In silico medicine, as one of the pioneer AI drug discovery company, published an article in Nature Biotech a few years back. It took the target DDR1 kinase from prior biological study. And in this article, they claimed that in 46 days, they were able to do the work that traditionally would have taken 1 or 2 years. Essentially, they took all of the target, the chemical compounds targeting the DDR1, and then form a database of chemical structures and feed it into a model. And this model will take all of the existing chemical compounds and hypothesize what pharmacophore of the protein structure they may actually be targeting to. And then generate a bunch of other molecular structures using a decoder and variational encoder architecture. And then all of these chemicals were further prioritized based on predicted properties, especially for the affinity of binding to the proteins. And eventually they selected one that they predicted to be better than the original DDR1 kinase inhibitor, and then put it into the pipeline and claim that they have validated. So this is a very interesting approach, right? So you generate new chemical compound using the language of the chemistry and you use the constraints of the compound's biological docking to actually help you guide the selection of chemicals. So at this point, it would be really nice to revisit that what are the different possibilities of using generative AIs, starting from even some of the archaic models. So actually in 1984, there's a rule-based expert system, and many people today don't believe that they belong to the new generation of AIs because it's based on expert system that have an inference engine, lots of rules. But they're very relevant to physicians because physicians think in terms of the facts from the medical text, and so are the biologists. For example, we may have a database that has A, B, and C. A is a protein X is expressed, B is oncogene Y function is attenuated, C is cancer cell growth stalls. And then you can express some rules. If A, then B, if B, then C, if you have an observation of A, then you have a conclusion of C. So essentially in this context, you can say if X is expressed, then oncogene function is attenuated. This may actually come from the experiment. That's how biologists are adding to the body of knowledge. If they observe this as a new novel regulator of oncogene, then in the database, and they said oncogene Y is attenuated, then cancer cell growth stalls. Then once you express highly A, which is the therapeutic methods, then you get the new knowledge, which is cancer cell growth stalls. Right? So even today, I think that this is still relevant. There's another model, and today we consider them not to be the true AI model. We call it the machine learning model. But this work published in 1992 is still relevant, right? So it's published by researchers from the National Cancer Institute. It's looking at the anti-cancer agent, right? In this particular example, I listed 4, and how they are promoting or inhibiting 60 different cell lines growth, according to NCI 60 cell line samples. The bars going left or right is essentially compared to the mean. Is it actually promoting growth or inhibiting growth of these cell lines? So each of the drug, you can obtain a profile. So the researchers look at essentially using a artificial neural network that's not a deep learning network, but it's a very shallow neural network. And essentially, they were looking at all of these chemical and then train the artificial neural network or machine learning methods to one of these categories of called the labels of these drugs and mechanism of actions. So some of them are alkylations and some of them are changing the topomerase activities. And when they look at, after they're done with the training, they look at the result of a prediction result of compounds. And most of the categories, then they were able to predict very accurately. So essentially, if you want to generate labels for the drug category, and even some of the simple method would work. Obviously, the Model 3 is very common before large language model come into existence. And any graduate student today in biomedical data science can run some data and through some pipelines using Jupyter Notebook, perform machine learning, deep learning classifications or some clustering. You can build a model specifically for a very specific task, right? And they're just not the generalized task. But it's good to actually refresh our memory about the first example that I mentioned, which is really about the symbolic approach to AI, right? So the first example is always reasoning using human symbolic language. For example, if you want to know what is an apple, you can look at the apples, this fruit's origin, is it coming from an apple tree or not? Look at the structure, does it have a body and a stem and different colors? And what's the classification from the label? And use the symbolic system to inference. That's actually the first approach, generative AI. And the second approach is the second example I listed before, is simply try to connect with internal nodes. It doesn't matter whether it mapped to the symbol or not. And these two approaches have not been just compatible with each other. So one is more interpretable but lacks performance. One has good performance but may not have good interpretability. To the point that if you go back 5 to 10 years, this is what we teach the students, integrating to a system. On one side, we need to have statistical machine learning, learn features and try to understand what the feature map to. And on the other side, and try to use a knowledge base, use ontology, and then you somehow blend it together to generate the knowledge. So this is the way that the biomedical research is done. It's very much of a based on well-calculated machine learning approach that does smaller granularity of work, and then try to use a human reasoning of biology to make sure that we're not making logical leaps that contaminate our knowledge. However, the larger language model really take us by surprise because it does seem to understand the language now. For example, it just uses deep learning and it uses human language. And here's a question. The woman went to the store and bought a what of shoes? Most of us may think it's a pair of shoes. And then you can also say a cart of shoes. And some women may have a shoe fetish. But then the large language model would be able to learn the statistic, maybe 80% of the time to say pair of shoes, and 20% of the time will say a cart of shoes. So somehow the ability to learn the context is exactly mappable to many of the biology that we understand. For example, in genetics, there's a key concept called a pleiotropic property of genes. That means a gene can actually have more than one trait, right? Exactly what trait it has depends on the tissue context. And even Mendel, in his original experiment, observing the peas in different generations, observed that the genes responsible for the seed coat color are usually linked to the colors of flowers and the colors of the plant axis. So you can think about this as gene having contextual information depending on the biological context of cell lines in animals and human may have very different functions. And this is what biologists today have to test it, one model, one cell line, one individual human at a time. But I haven't seen anyone to actually do it in the large language model context, and I predict this is upcoming, right? And underneath the hood, there's a transformer that have self-attention mechanism that really is generating the contextual understanding of the word. For example, there are two sentences that are almost the same. The animal didn't cross the street because it was too tired, and the animal didn't cross the street because it was too wide. Both term it, you need to understand what it really refers to. Right? Self-attention is actually blanking it and try to learn what is the weight contribution of each of the word to the word it. And in this particular case, it correctly, through self-learning, understanding this it refers to the animal and this other it is the street. So imagine if we have gene expression and functional context of the cells, and wouldn't it be nice that we can develop a large language model for understanding gene function, or especially the pleiotropic gene function, right? So there's exponential growth of LLMs, and many of you already know that over the years, and this growth of the size is exponential, largely thanks to the arrival of GPUs and lots of cheap computing power. But our power usage has not been a problem that has been solved. And that is the future bottleneck of using it in an everyday biology setting. But nonetheless, the bigger the model, usually the bigger the performance. And then the trend of generative AI model is to make the generative AI model self-supervising. That means that you only need a few instances, not a lot of instances, and then to succeed, and multimodal with many, many parameters. So the data modality, as I mentioned in the very beginning, can take genomic information and many, many types of data information, including voice, images, videos, and then there are many applications, and I wouldn't go through because some of them has already been covered in the previous LLM lectures. But I do want to mention that the multimodality artificial general intelligence many people predicted is actually was just launched by Google earlier this month, and it actually supports multimodality, just generative models. Your input sequence could be any combination of these text, voice, image, and video, and it goes through the transformer, and then it will be decoded into image and text for now. There's no video or voice, but I'm pretty sure in the future they will add it. And especially when you look at the Gemini 1.0 performance, according to Google, obviously I did not do the benchmarking, and it outperformed GPT-4 in almost all the categories except for multiple choices. And this is really astonishing. The reasoning capability is better, and the math capability is better. And it's not just the improvement on the size. So if you actually look underneath the hood, the Google is using a new architecture that's based on not in the NVIDIA's power-hungry GPUs. It's using TPUs that's very much of energy efficient. And then they were able to scale up the learning of images and videos in ways that GPT, even OpenAI architecture, is unable to ingest, right? So if people have OpenAI have unlimited, meaning even tens of billions of dollars, they would have done it. But ingesting videos would requires very careful design of hardware that is power efficient, that can also have the enough bandwidth to learn this. But they were able to achieve that. And here's an example, right? So they show the example, write code to arrange these figures and with some constraint. Put this to the first position, put this to the last position, and then for the other two, fix one, and then fill in the remaining one. So it's a logical trick. See if the AI could do it. And also, It says that you have to explain and then write a code so that you can generate this. So Gemini 1.0 was able to respond with the right code and get all of the answer and generated a figure in the end. So this is very impressive, right? Because it's a truly multimodality AI. Imagine this is a pathology image and some instructions or some gene expression profiles, and it's able to generate this gene expression profile along with some descriptions. And then you can start to see the true power. And then there's another video.
A common problem scientists face is the need to find and use data extracted from the scientific literature. This is difficult because scientists need to search among thousands of scientific papers for key information and extract them by hand. It's a very common workflow and very time-consuming. In fact, some of our scientists at Google DeepMind face this very problem. They use Gemini to help with it because Gemini has an incredible understanding of science. Taylor will explain more.
So we were looking at this study from 2022. The authors had created a data set by reviewing tens of thousands of scientific papers in genetics. They found a few hundred papers that contained the relevant information, extracted it by hand, and collected it in a table. Studies like this can take a lot of time. We needed to update this data set with what's new over the last couple of years. But that's over 200,000 new open access papers added to this domain since 2021. We couldn't do this manually, so we asked Gemini to help us out. First, we needed to filter for relevant scientific papers. We wrote a prompt, just like this one, telling Gemini exactly what to look for. With its advanced reasoning capabilities, Gemini was able to distinguish between papers that were relevant to the study and those that weren't. For the relevant papers, we wrote a similar prompt, asking Gemini to read the paper and extract the key data for us. We could even ask Gemini to add annotations. This showed us exactly where in the paper Gemini found the information. We ran this at scale. And over a lunch break, Gemini read 200,000 papers for us, filtered it down to 250, and extracted their data. So now we have a refreshed version of this data set. But because Gemini is multimodal, not only can it reason about information from text, it can also reason about figures. So let me show you something really neat. With our refreshed data set, we can now ask Gemini to update a graph from the original study. We first gave Gemini a screenshot of this figure. Then we asked it to generate the code required to plot it. And by feeding this code our new data set, we get our updated figure. You can see that this figure now includes data up until 2023.
So Taylor used Gemini.
Okay, so that's it. I think I was very impressed, and it's almost like a month of my graduate student work in 1 hour. So I think now it would really be good to think about how do we prepare for disruptive revolution if we can do generative AI for translational biomedicine. So I put in the checklist, right? So I think first of all, the environment has to accept the AI, generative AI application in your area. If your collaborator doesn't want it, and then don't try it. NIH doesn't want it, and then don't do it. But then I think you want to really make sure that this generative AI is generating new knowledge, not hallucinations. And it would really have the human health impact to outweigh the potential burdens of setting things up. Because if you don't have a specific mission, maybe the general ChatGPT refinement will work. And then you have to go through some technological breakthrough and get the right data to train your model. And you have to have the right computing power and lots of collaboration. So I'll just quickly go through this since I only have limited time, 10 minutes left. In my group, we've been preparing AI-ready data for drug discovery for a long time. So we have 56 million drugs captured from various sources, 700,000 therapeutic methods in clinical trials, drug-to-drug, disease, and gene-to-disease information that are all categorized into databases. And that's step one. And there are actually even founders of Facebook. There are also funding initiative that actually will map between cell and the gene. For example,
we are really optimistic about the potential for AI to help scientists cure, prevent, and manage all diseases in this century. Now, one of the first problems that AI can help scientists understand is the intricacies of how cells work, both individually and together to make our organs and bodies work. So one of the first AI projects that we're going to work on, we call the virtual cell. And it's going to help scientists map out all of the different states that all the different kinds of cells can be in, both when they're healthy and diseased. And to do this, we are building out one of the largest AI compute clusters in nonprofit life sciences research in order to train models like the virtual cell.
And to do this type of AI research, we're going to need a lot of data. At CZI, we've been building up to this moment. We funded scientists to generate single-cell data sets. The San Francisco BioHub has completed whole organism atlases. And tools like Cell by Gene have helped scientists standardize and aggregate data. We won't be able to do this alone, but together with these advances, we're going to be able to move the needle to cure, prevent, and manage all disease by the end of this century.
Yeah, so I think we know that as an bioinformatician, we know the cell by gene is nothing but actually getting all the different cell types of a particular disease or health state and mapping out all the gene expression. So NIH has already done many projects. In fact, my team, along with Trey Edgar and at UCSD and Stanford, we're onto something even bigger. We call it Cell Models for AI. And there's a website, CM for AI, if you're interested. We're going to have the first major data release next March. Essentially, what we're doing is to look under the subcellular level, looking at different induced polypotent stem cells, cancer cell lines. We're looking at PPI information, immunoco-participation information from immunostaining microscopy information. And then we do genetic perturbation. And we not only present the data, but also assemble the data in a hierarchical structure such that people can not only use this data, but it can also do explainable AI on top of it. So things are moving. And in my own research group, we also have an innovative standardization platform that makes such gene expression data AI ready. We call it the gene terrain. So if you have genes and sample, that sample could be cell lines, could be individuals, and we can actually make digital images. We have paper, I forgot to actually mention the paper reference here. So essentially by dialing the parameters, and we can actually turn the gene expression information into X-ray or functional MRI-like images. And then you can apply all of the interesting image processing and AI techniques that work natively against the images, as opposed to hard to parse gene expression information. So we have shown that you can actually use this technology, look at the 26,000 cells from 11 glioblastoma patients. And by aggregating cell patterns, gene expression patterns, you'll be able to see either very similar patterns or very dissimilar patterns. And that has a lot of implications. I think that's our effort in making the data AI ready. So I also want to mention that in this area, the industry is leaping ahead. Right? For example, NVIDIA has BioNeMo LLMs for biology. It has LLM that can actually allows you to understand the protein sequence structure. It would generate representation of protein sequences. It would also have the structure, help you understand the protein structure, help you understand the chemical compound. The only thing it doesn't do is what we care about passionately is the cell regulation, gene regulation information, right? I'll skip some of these, and I want to actually mention that in this context, we formed the System Pharmacology AI Research Center. We want to curate AI-ready open data set for drug discovery, where everyone can come in and make use of and hopefully contribute. We want to benchmark AI algorithm and generative AI platform to say which one is better for which purposes. We want to augment teams who are interested in using AI augmented method to maybe do drug discovery. And then we also want to promote the academia industry partnership to the point that we can educate the public about the emerging AI-generated efficacy and safety issues. Okay, here's the website. We're gonna have a symposium at UAB if you're interested. It's April 28th, and let me know, and I may actually put you as a speaker. But also in the last, I would like to make one final pitch that I think it is a time that we start to think as a community about the Manhattan Project equivalent, which I call the Generative AI Model for Translational Medicine, GAM4TM. And the GAM4TM's goal, in my opinion, is to actually use comprehensively our resources from the government and institutions, pull together expertise so that we can develop a LLM or generative model to master the language of the cells, genetics, human biology, along with a human language, so that we can interrogate this model and get biological insights and directly maybe accelerate the understanding of human biology to disease cure. To do that, and I think we have certain components already, I won't go through the detail since I'm running out of time, but this is my last slide. My personal outlook for GAM4TM, there are challenges, right? This would require 10 times or 100 times more effort requires to achieve the artificial general intelligence by resource-rich companies. We have to get the data AI ready. That takes a lot of effort. We have to really overcome the multi-scale complex human biology and health knowledge representation problem. And we are lacking the computing resources. Most of all, this is human subject study, but the opportunity is huge. If we can all come together, collaborate, and if we can convince the government today, starting from today, that we need such a tool, just like a James Webb Space Telescope that takes $1 billion, and then we can innovate AI, genomics, and biotechnology. We can make sure that it's trustworthy and benefit all of us. With that, and I'd like to thank the people and take 1 minute of questions.
Thank you, Professor Chen. This is fantastic lecture. We are going to take questions, and you can put your question in the Q&A chat. I'm going to invite Alexander to help me and offer the questions to Professor Chen.
Yeah, I can start with the type of question. Do you think there's value in accepting the reality student will use the tool to cheat and then adopt by making the tool grow and develop hands-on project? Yeah, I think I'm already encouraging students, including my 11-year-old daughter, to embrace the technology. But I think there's a pros and cons. I remember I grew up in a culture of Chinese culture where they use abacus and they pride themselves in parallel computation using 10 fingers to calculate on the abacus, but then it's going to become a specialty. So if you want to actually become more able yourself and in your sharp mind, in your reasonable capability, you don't want to lose the opportunity to train your student to develop their own reasoning skills. But if you're more concerned about the productivity of the project, sure, I don't see a problem with that. Way to learn the success of generative AI and text images. How do you imagine research on biomedical modality other than text and images can be benefited from the AI breakthrough? I think this is a very good question. I don't put claim to actually be the leading innovators and the fundamental researchers to really train this generative model with, let's say, videos and voice. But I think there are people at the Google and Amazon OpenAI, they're doing fundamental work. We usually keep track of the paper that they publish and see opportunity to make it more efficient. That's where we can contribute. There are many LLM models available, open source and so on. To try LLM for biomedical research, which model would you recommend to start with? It really depends on your purpose, right? If your purpose is to use human language interface, I think a ChatGPT, GPT-4 is still the best. But if you're really wanting to train with your specific data set that is not in the public domain, you actually have to use GPT-4, certain functionality that you can actually fine-tune the model. Or if you want to rebuild the model from scratch, then you have to use the open source one. But that's all assuming you have the computing resource. If you don't have the computing resource, then your only option is the Google Geminis and the Bing and the commercial available models. You like the GAM for TM proposal? Thank you. Do you think it will be possible given all the private entity all racing to achieve such a goal? I think it's possible because this GAM for TM is actually not for profit. We're doing this for the curiosity of the human knowledge. It takes a private foundation such as the Chen Foundation to do a small piece of it. I don't think Gates Foundation's focus is in here. Nobody have the deeper pocket of NIH. So if we can all maybe write to me and then write a white paper to pitch to NIH, perhaps we have a chance. There's probably very little commercial values unless we want to make drug discovery. That's obviously that's a small part of the bigger model that I'm proposing. Exciting about the CM4 AI. Good. Will that be shared in a way that's accountable? If you go to cm4ai.org website, you can stay tuned. We will have a Jamboree. I'm not sure how open that Jamboree will be. We will actually release part of the data and tools for people to explore. We're curious about working with Gemini. Presently, Google only allow access to a specific partner. Do you know any group? No, I don't know any group. I have a personal friend working at Google, but I don't think we can use the backdoor for official collaborations.
Okay, looks like we've gone through all of our questions in the Q&A. Last chance for anyone to ask some questions.
And if you're still collecting your thoughts, please do put the questions together. We have a way of forward your questions to Professor Chen for his response at a later time. Again, please join me to thank Professor Chen again for such an innovative and insightful lecture. We are very grateful. This particular lecture, together with all other large language model lectures, as well as AI ML lecture series, are currently available on Bridge to AI Bridge Center program website. Thank you again for joining us. Thanks, Jake. Thanks, Wei. Thanks, Alexander. Thanks, Tim.
Thank you.
Bye-bye.